afternoon, you've joined us live at UWS Labs, where we've been following the mysterious crime of who poisoned Dave Angel. We're minutes away from hearing the verdict on the suspect, but first let us hear from the scientists who investigated these findings with some extensive research and experiments. Dr. Pavlova, first of all, could you tell us why you had to complete the solubility test?
Okay, so knowing that some sugars are soluble in water, as well as some amino acids and proteins, this helped identify what type of breakfast could have contained the poison. To match the contaminated breakfast description, we were looking for suspect powders which did not dissolve. This means that the breakfast likely did not contain much sugar or amino acids, therefore we were able to rule out the other breakfasts.
That's so interesting, thank you for that. Now can you tell us how you tested for this?
So to test for solubility, a heaped spatula of each suspect sample was placed into a boiling sheet with 10 ml of distilled water. Each suspect powder was then mixed and recorded the length of time it took to dissolve. Referring back to the biochemical analysis of the contaminated breakfast, it was shown that we were looking for the suspect sample which had a low solubility. This ruled out suspect sample A and D as they were easily soluble. Suspect sample E was also soluble, however it took much longer than suspect A and B and therefore was not ruled out for the solubility characteristics. Suspect samples B and C had low to none to no solubility and therefore have the closest solubility characteristics to the contaminated breakfast.
So how is this relevant to the suspected poisoned breakfast?
Okay, so referring back to the biochemical analysis of the contaminated breakfast, it was shown that we were looking for a suspect sample which had a low solubility. This ruled out suspect samples A and D as they were easily soluble, and suspect sample E was soluble but took much longer than suspect samples A and B. Therefore, was not ruled out for the solubility characteristics. Suspect samples B and C had low to no solubility and therefore have the closest solubility characteristic to the contaminated breakfast.
Thank you for that, Doctor. Okay, Doctor, so you tested for vitamin C. Do you mind just explaining to us why?
Yes, so vitamin C is important as it is found in many citrus fruits and vegetables such as limes, oranges, tomatoes and green peppers. It is very important for a healthy diet and maintaining healthy connective tissue, which can prevent a person from developing scurvy or other potential health problems.
Okay, Doctor, so can you tell us how you actually carried these tests out, please?
Yeah, so to test for the presence of vitamin C in the subject samples, we use DCPIP, which acts as an indicator. DCPIP changes colour from blue to red with the presence of acids and will lose its colour in the presence of certain chemicals such as ascorbic acid, more commonly known as vitamin C. For the DCPIP test, we are looking for our samples to change from a red colour to a blue colour. As you can see from our results, samples A and E showed a positive result for the presence of acids. With further testing, we were able to identify that no samples had the presence of vitamin C as none of them changed to colourless. Another test which was carried out was the litmus paper test. By using red litmus paper and looking for changes to blue, we were able to identify that none of the suspect samples were of a basic functional group. By using blue litmus paper and looking for changes to red, we were able to identify that suspect samples A and D were both acids. As suspect samples B, C and D had no reaction to either of the litmus papers, we are able to say that they have no acidic or basic functional group present in the suspect samples.
That's great, Doctor, thank you for that. Could you tell us how that's relevant to your suspected breakfast?
Yes, as the contaminated breakfast did not contain vitamin C and all of our results also did not contain vitamin C, we were unable to rule out any of the suspect samples, therefore further testing had to be completed.
Okay, that's great, thanks for your time, Doctor. Okay, so you've just heard there how solubility tests might be relevant to the suspected poisoned breakfast, so we're going to move over now to Dr. Ritchie. Doctor, you're going to speak to us about fats and lipids, but could you explain to us first of all what fats and lipids actually are?
Diana, of course. So as part of our investigation into who killed Dave Angel, we tested the suspect breakfast for the presence of fats and lipids. Now, fats and lipids are a group of macromolecules of slightly varying structures but similar properties. They are complex polymers featuring one glycerol molecule and a variety of fatty acids. The fatty acids are long-chain hydrocarbons featuring a carboxyl functional group, and this gives them certain properties which we're able to test for to show their presence. Animal fats, for example, contain triglycerides, which is one glycerol molecule and 3 fatty acids. Excellent storage for long-term energy because of the high proportion of carbon and hydrogen bonds there. In foods, you'd find them usually in animal produce, such as meat products and dairy, and that will help us to determine which of these breakfasts kills Dave Angel.
The results of the emulsion test can be seen on screen. Samples A, B, C and D were seen to turn cloudy, indicating the presence of lipids. Sample E did not contain lipids.
A second investigation that we carried out to determine the presence of lipids and fats was a greaseproof paper test. So it's a very simple test. We took our 5 solutions of breakfast samples, 2 controls, one of oil and one of distilled water, and we placed these onto greaseproof paper. When these are dried in the oven and then shown against the light, you can observe translucent grease marks. Now, if there were lipids present in the samples, you would see these translucent grease marks. Samples that did not contain lipids would show no difference.
As can be seen from the results of our grease paper test, only samples B, D and E were shown to produce translucent grease marks, indicating the presence of lipids within those samples.
Our third and final test to determine the presence of lipids in these samples was the Sudan 4 test. This is a red dye that's fat soluble, so it's selectively absorbed only by lipids. If we mix this with each of our solutions and our 2 controls, and there were lipids present, it would show as red globules either on the surface of the sample or suspended within it.
As can be seen from the results of our Sudan 4 test, sample A and sample C were seen to produce red globules, indicating the presence of lipids.
Breakfast. Of course, so as seen in our results, sample C contained fats and lipids. In 2 of our 3 tests, it was shown conclusively to contain fats and lipids. Now, the breakfast that killed Dave Angel was known to contain fats and lipids, therefore it can be deduced that sample C may have been the breakfast that killed Dave Angel.
Okay, thank you, Doctor. Okay, that concludes our first part of our interview. Next we'll be speaking to Dr. Kerr and Dr. Lanigan on their findings to this poison breakfast. Hello and welcome to part 2 of our interview to find out who poisoned Dave Angel. I'm now joined by Dr. Kerr and Dr. Lanigan. I'm going to ask how they got on with their experiments and findings. So, Doctor, could you please explain to us what proteins are, first of all?
Absolutely. Proteins are a very important group of macromolecules to cells due to a large range of cellular functions, such as enzymes, which facilitate specific chemical reactions, and cell defence, where proteins use their shapes to detect foreign pathogens, such as cancer cells, or even in nutrient transport within cells, using hemoglobin for an example, the protein responsible for carrying oxygen in red blood cells. Proteins are large structural molecules made of amino acids. Each amino acid features an amine group as well as a carboxyl group. These amino acids join from the carboxyl group to one amino acid group only by a peptide bond.
Okay, thank you for that, Dr. Kerr. Now, I believe you carried out some tests for this. Could you just explain what you did?
So we carried out 2 tests, one to test for proteins themselves and another for amino acids. The first test we carried out was the burette test, which was the test for the proteins themselves. For each of the 5 sample substances, a solution was prepared using a sample of said substance and some distilled water. Dilute copper sulfate was made by adding 9mm distilled water to 1mm of failing solution A. 6 boiling tubes were set up, each containing their own sample, one for control with just distilled water, and 10 drops of dilute copper sulfate were then added to each boiling tube. 2.5 ml of 40% NaOH was then added to each boiling tube, and after a short while, the boiling tubes were observed and showed colour changes depending on the presence of proteins.
Results from the burette test show a very positive protein presence in sample B, a lighter presence in samples A and D, but little to no difference to the control in sample C. The second test we carried out was the ninhydrin test, which was used to identify amino acids and will therefore give a result for present proteins as they are built of amino acids. Like the burette test, this test featured a control in the form of distilled water as well as our 5 subject sample solutions. 6 far separated circles were drawn on a piece of filter paper using a pencil. A drop of the samples were added to their own labelled circle on the paper. The filter paper was then briefly let to dry before a drop of an anhydrine solution was added on top of each circle. The filter paper was let to dry for a longer interval of time for the results to show. A colour change to violet from colourless indicates a positive result for amino acids. As seen in the results, all tested samples showed a positive result for amino acids as shown by the purple colour change. This is why 2 different tests were carried out. Just because amino acids are present doesn't mean proteins will be too.
Thank you, Dr. Kerr. So you just heard here how Dr. Kerr explained how proteins are relevant. Now we're going to cross over to Dr. Lanigan. Dr. Lanigan, could you please explain to us what carbohydrates actually are?
So carbohydrates, also known as saccharides, are molecules made up of carbon, hydrogen and oxygen held together by chemical bonds. They make up staple foods like bread and pasta and are key for energy in humans. Now carbohydrates may be monosaccharides, mono meaning one, and they're like the simplest form of carbohydrate, so that's like fructose, glucose, for example, but monosaccharides can join together to become disaccharides or polysaccharides, more like complex, like starch. Now, disaccharides and polysaccharides, they act quite differently to monosaccharides. As I said, they are complex, which is why we needed to perform multiple different tests for carbohydrates.
Okay, doctor, so we're going to talk about the tests that you carried out and what they meant. Now, I believe that the first one you done was called the Fellings test. Could you explain a wee bit more about that, please?
Yeah, of course. So the Fellings test identifies simple monosaccharides like fructose and glucose. They will give a positive reaction to the test. So the experiment has 2 versions because the solution needs to be hydrolyzed in order to get the results. The test is carried out by getting a sample of the breakfast, putting it in copper sulfate solution and sodium potassium tartrate, and then placed into a water bath for around 10 minutes. And then if the solution turns a bright red colour, then it's positive for the simple sugars, so we know that they are contained in the sample.
As you can see from our results, both samples C and D changed to a brick red colour, indicating the presence of simple sugars.
Okay, Dr. Lanigan, if you could please explain to us how you carried out the iodine test.
Yes, of course. So we carried out a separate test from the beginning to test for starch, because starch is a more complex sugar than the monosaccharides for the Fellings test. This is because it is a molecule that is coiled, it is arranged differently as it is multiple fructose molecules put together. So for the iodine test, we took the samples of the breakfast, added 3 drops of iodine to them, and then mixed them with gentle agitation until they turned a different colour. Now, if the tests were positive for starch, they turned a bluish-black colour, and if they never, they went a yellowish-brown.
As you can see from our results, samples A and E never changed colour, meaning there was no starch present. Sample B changed to yellow, also indicating there was no starch present. Samples C and D both changed to a dark, almost black colour, indicating there was starch present.
Okay, doctor, so can you tell us, just in conclusion, what this means?
Well, since we know that the breakfast that Poison Dave Angel definitely contained carbohydrates, it was really important to test for them. As you can see from our results, sample C contained them both times, so that really furthered our suspicions that C could be it.
Excellent, doctor, thank you.
This investigation comprised a series of experiments designed to determine the presence of certain macromolecules within samples of 5 different breakfasts, each suspected of poisoning Dave Angel. The aim of this investigation was to build a biochemical profile of each sample in order to ascertain which of the breakfasts had been contaminated. The results of these investigations are recorded in the table shown. What conclusions may be drawn from these results? When comparing the macromolecular composition of each sample with that of the breakfast known to have killed Dave Angel, sample C was found to be the closest match, suggesting it was a meal which had indeed been contaminated. As demonstrated by the results of these experiments, sample C was shown to contain no glucose or fructose. However, the sample tested positive for the presence of sucrose and lactose, starch, protein, and amino acids. And this was also the case for the breakfast known to have killed Dave Angel. Furthermore, neither sample C nor the breakfast responsible for the death of Dave Angel contained vitamin C
and both tested positive for the presence of fats and lipids. The solubility for both was found to be very low, and using this knowledge, it was possible to compare this profile to the breakfast menu of the week. Monday's offering of yoghurt and orange juice may be ruled out as the killer breakfast on account of the presence of vitamin C in orange juice. Tuesday's option of scrambled eggs doesn't appear to have been served with any form of carbohydrate. Knowing that our sample tested positive for complex carbohydrates, we may rule this meal out as the poisoned breakfast. Thursday's breakfast of porridge oats with milk and honey topped with fresh raspberries also does not fit this profile, as it will contain glucose and vitamin C. Friday's breakfast of sugar puffs with whole milk may also be eliminated for the same reason that it contains glucose, whilst our samples were shown to contain no glucose. And so, by process of elimination, it can be determined that the breakfast that killed Dave Angel was Wednesday's breakfast, the best butty, locally produced pork sausages in a morning roll. This matches our profile, as it contains complex carbohydrates, proteins and fats with no obvious source of vitamin C. Knowing that Dave Angel's breakfast was poison on Wednesday, it was possible to identify the waiter on shift that morning. The waiter responsible for poisoning the breakfast of Dave Angel was none other than Alexander.
Okay, so you've heard it here first. 5 breakfasts, 5 waiters, 5 days. The conclusion is breakfast C is contaminated with all these things.